Hi, this is a short video covering some of the key concepts from our practical class on cardiovascular disease, histopathology. We talked about Joe, and we started with the problem of atherosclerosis that Joe had in his coronary arteries. You remember we saw a medium-sized coronary artery with eccentric intimal thickening. And we also saw that coronary artery that atherosclerotic plaque had been complicated by the process of thrombosis. You remember thrombosis is coagulation occurring in a vessel during life. That thrombosis had led to decreased blood flow, leading to tissue hypoxia within the heart, and the term we used to describe that was ischemia. Decreased oxygen in the tissue because of decreased blood flow. When that ischemia continues for some time, it may become irreversible, in which case we have an infarction. An infarct being a localized area of ischemic necrosis. And that brought us to our next slide, which is a slide of myocardial infarction, a piece of potentially of Joe's heart, and we saw within that slide we talked about the clinical issues arising from that and the pain that Joe would have experienced because of both ischemia and infarction. And we highlighted at a histopathological level the problem of coagulative necrosis. Coagulative necrosis representing the morphological changes of the process of infarction, at least in this tissue. You remember we found three key features in coagulative necrosis at a microscopic level: loss of nuclei, increased eosinophilia of cytoplasm, and preservation of the shape of cells and tissues. The next problem that arose, the slide we saw, was that of a renal infarct. And we talked for some time about how that could have arisen, and we realized eventually that the issue here was that there was thrombosis, that same process occurring on the left ventricular endocardial surface, a mural thrombus. That was a complication of myocardial infarction, and that had embolized a thromboembolus in the systemic arterial system that had lodged in a branch of the renal artery on one side and produced again a localized area of ischemic necrosis called a renal infarct. Here clinically there's a quite different thing. It was causing hematuria, the presence of blood in the urine, but microscopically the same process of coagulative necrosis was seen. Same morphological feature telling pathologists that this is an area of infarction. The next slide we saw was again related to that same phenomenon. Again, thrombus on the left ventricular wall, a mural thrombus breaking off and embolizing to form a thromboembolus that lodged this time in the cerebral vascular arterial system and caused a localized area of infarction within the brain. That should be cerebral, not cerebral. And that localized area of infarction produced obviously significant clinical effects in Joe, which we refer to as a stroke or a CVA, cardiovascular accident. This time, the changes were quite different morphologically. The brain actually dissolved and underwent liquefactive necrosis, unlike coagulative necrosis in other tissues, but it was still the process of infarction. Finally, we looked at a slide which was another example of thrombosis, but this time thrombosis occurring not in the arterial system as all the others had been, but in the venous system, and brought about, no doubt, by bed rest and immobility consequent upon Joe's stroke. Again, we highlighted the idea that thrombus in a deep vein of the leg could have, or indeed in Joe's case did, embolize, break off. I'm sorry, embolism should be EMBOLISM there. Did break off and spread, in this case, a systemic venous system travelling back to the pulmonary arterial tree, causing a pulmonary embolism. And in Joe's case, because this was a very large pulmonary thromboembolism, It impacted centrally within the pulmonary tree, arterial tree, and caused sudden death. That's the story. Hope you got all those points out of it. If you didn't, you should consider also your lecture notes on this topic. Thanks.